going to go through some fun things like diseases, microorganisms and disease as that relates to human beings. We're going to go through some microbial ecology, which we're starting today. How do microbes interact in the environment, and why do you care about them beyond whether or not they're causing a disease? They're hugely important to this globally. So many things in life that you just take for granted are microbially driven processes, and we're going to go through that a little bit today. But also we'll get into a little bit of disease. I have rearranged your schedule just a bit because of the storm, and so I apologize. I think what we're going to cut is immunology. I'm going to cut the immunology lecture, and that's a shame because I love that lecture. It's fun, it's tough, but it's so important to so many of you because so many of you are pre-dental, pre-med, pre-vet, pre-something like that in the health professions. That it's a good introduction to that is something you need in your curriculum, and that's why I've always kept it in this class. But something has to go because we missed an entire week of class. So we will miss that, and then unfortunately the review session will probably get cut a little bit short at the end of the semester as well. And I'm really sorry for that. That was unavoidable circumstances. So let me do all that technical stuff after I lecture. And I'll get through the lecture for a little while, and then we'll make sure I have plenty of time for everybody to look through their exams, and we can talk about this semester. So what we're starting with today is kind of getting into the ecology portion of this class. How do microbes interact in the environment, and how do they interact with other organisms? Obviously, you spend a lot of time thinking about how they interact with yourself. They're interacting with my head and my nasal passages as we speak. But they also have a lot of really important environmental jobs in terms of things like respiring oxygen that we all need to breathe, and decomposing dead things, and cycling nutrients in a way that keeps life going on this planet. I argue is all microbially driven. It's all at its core carried out by microbes. So, microbes have been on this planet for 3.5 billion years. You learned that at the beginning of the semester when I showed you the timeline. We've been evolving for a really long time and making profound changes to ecosystems. As I just said, nutrient cycles carried out by microbes are what make life possible. So one of my favorite anecdotes for this class that people don't ever really think about, but whenever I say it, a lot of people go, oh yeah, of course. We're so obsessed right now with things like climate change and increases in CO2 concentration in the environment. But when you look back through the history of this planet, probably the #1 biggest pollution event that ever occurred on this planet was photosynthesis. So the advent of photosynthetic cells that figured out how to capture carbon dioxide from the atmosphere, break a water molecule to exploit that energy, and then what did they do? Release oxygen. Absolutely. So it's kind of microbial ecologists like myself like to throw that out there and kind of remind everybody, put things in perspective a little bit. That we wouldn't be here if a couple billion years ago microbes hadn't figured out to split that water molecule, accidentally releasing a bunch of oxygen as a waste product, oxygenating the planet and allowing aerobes like us to thrive. So when we think about their importance in terms of global cycling, keep that in mind. It's really, they're very central to so many things we completely take for granted. And let's define biogeochemical cycling. This is a huge word. If you haven't heard it before, it's a stressful word, but not really if you break it down. Okay, cycling, turning something over. Conversion in a not in a linear fashion, but in a cyclic fashion. Biogeochemical, so chemical. We're cycling some sort of chemicals. Geo, earth. So we're cycling the chemicals associated with earth. And bio, it's a biological process. So the biological cycling of earth's elements basically is what biogeochemical cycling is. And what we're going to work on today, obviously this is not just a microbial process. We're all sitting here right now respiring CO2 out and taking oxygen. And trees breathe, etc. But so much of it is microbial. And the things we're going to talk about today, I'm going to focus on the microbial aspect of that in terms of their different roles. All right, so this is a cartoon cartoon. Some of you have seen it before. But let me be clear. So some of the, from other experiences with students, I want to make sure you understand this is not a microbial nutrient cycle, is it? No. This is a rabbit and it links or something, it's a big tree. Not really microbial stuff. The only part of this that's really microbial is right down here, at least in this picture. But the reason I put this picture in is because I want you guys to start getting a feel for the way we think about flows and cycles. So far in this class, we spent a whole lot of time talking about the minutia, talking about literally an operon. And if one cell is exposed to lactose, what gene does it turn on? But now what we're doing in the next couple of lectures is really expanding that out to think about almost at a global level, or certainly an ecosystem level. And most of you in this room have had your ecology already. If you haven't had your ecology, you haven't had a couple of you have. All right. So if you get confused, just reel me in and I'll fill you in. Ecosystem level meaning, you know, a whole huge suite of interacting organisms in the environment. And what's the microbial role? And so I want to get you guys used to thinking about that and the way that nutrients move through these things with this nice friendly little cartoon, pretty macroorganism. So what this is indicating is the carbon cycle. We're going to spend a little bit of time today on the carbon cycle and a little bit of time on the nitrogen cycle. So this is showing the carbon cycle. And the carbon cycle starts in an inorganic form. I like to start all cycles in an inorganic form. And the inorganic form of carbon that we care about is CO2, carbon dioxide. Absolutely. And carbon enters ecosystems from inorganic form to an organic form through this lovely process that we all take for granted, photosynthesis. In this picture, it's being done by a nice big pretty tree. But most photosynthesis on this planet is being carried out by a teeny tiny little floating cell out in the middle of the ocean or linkers. All right. So photosynthesis takes that carbon dioxide and converts it into sugars and then ultimately into amino acids and proteins and nucleic acids. All right. So then carbon moves through ecosystems because it gets trapped into an organic form through some kind of a plant life. It gets consumed by a primary consumer, in this case a little bunny, and then eventually gets eaten by the guy that eats that bunny. All right. And then CO2 returns to the atmosphere because those plants also respire. Okay, they break down those molecules and use them for energy the same way we do. And so does that lynx, and so does that hare. They respire that CO2 back. But also, eventually, everything dies. Everything dies and goes back into what we call detritus, or dead stuff. All right? And then microbes and others, fungi, bacteria, a lot of protozoa, will break all that down and respire that back as CO2. And this is where we're going to spend most of the lecture today. So this is just giving you a nice, easy look at how these things flow through systems. Hopefully, you're all rolling your eyes at me right now. This is, ninth grade biology. Let's spend a little bit more time down there. And then obviously the burning of fossil fuels is destructive of carbon cycle, which we'll talk about a little bit more in this class. All right. So forgive me, I didn't get this into your PowerPoint slides on Blackboard. I didn't do that. So this is a friendly reminder of what is this? Redox what? And those of you in my other class are sweating a little bit, you know I'm looking at you. I'm trying not to look back at you. Redox, what did she mean by redox? Redox potentials. And so what do I mean by a molecule being heavily reduced? At least one person honestly said. Heavily reduced, you guys have had chemistry. Start shouting it out. At this point, hey, you can be wrong and you'll still be a good answer.
Getting more double bonds, some single bonds between carbon and hydrogen.
Nice. What were you going to say?
Getting more electrons.
Gaining more electrons. More hydrogen atoms attached to it. Absolutely. More energy. Good.
Mediocre.
give up those electrons or gain more electrons when they're already highly reduced?
Give up.
Give them up. Good. Okay. So what this scale shows you is basically it's a really nice gradient in electropotential. The potential to gain or lose an electron. And electrons you can think of as energy currency. All right? Think of it as like dollars. And a cell that has a whole lot of electrons on it has a whole lot of energy and is nice and rich. And a cell that's been highly, what's the opposite of reduced? Good. A cell that's been, a molecule, an atom that's been heavily oxidized, it's losing those electrons. It's lost all those electrons, has less energy currency. So when you look at this, something that has a very, very negative E sub O means it's very, very reduced. And when it's very, very reduced, that means it has a high probability of giving up those electrons. There's a lot of energy being held in that molecule, and it has a high probability of giving up those electrons. Something like a hydrogen molecule, a glucose molecule, NADH should look familiar to you, FADH2 should look familiar to you. Those kind of molecules that are highly reduced are very likely to give up their electrons. And when they give up their electrons, they're transferring that energy to something. And the farther they go down that chain, the more energy that gets released with that transfer. So, if I dump my electrons all the way down on something like oxygen, a whole lot of energy is released. And that's exactly what you and I do. All right? We take in something like glucose, you eat a bagel, you take in something like glucose, and your body metabolizes it, and on the electron transport chain in your mitochondria, you dump all those electrons on oxygen. All right, so this should be a review so far. We're going to kind of kick up how impressive it is.
Bless you.
This is a review. You've seen this slide before. With the depths of your mind, you remember. You've seen this before. All right. Microbes don't always do something as simple as eat a bagel and then dump their electrons on oxygen. Absolutely, sometimes they can. Sure, it happens, but they can do a whole lot of other stuff too. They can use a whole lot of other different kinds of energy sources. Lactate, acetate, succinate, those are all sugars. And then the interesting thing about microbes that's going to become important in a couple of slides is they can dump their electrons on alternate terminal electron receptors, something besides oxygen, such as, so take a look up here and tell me, where else can you dump an electron besides oxygen and it's still energetically favorable?
Nitrogen?
I'm sorry?
Nitrogen?
Absolutely. What form of nitrogen?
NO2?
NO2 will work.
Mm.
It's not as good as NO3, but yeah, it'll work. Okay, if you dump your electrons on NO3, you'll end up with NO2. But you can also dump your electrons on NO2, so it's not probably good. Okay? Yeah, he's right. So also things like oxidized iron, okay? Things like that. What's not on here? Sulfate, things like that. Organisms can respire to sulfate. But relevant to the next few slides is the dumping of electrons on things like nitrate. And that's going to come through, because I want you guys to get just your first look at something called nitrogen cycle. I'm just going to give you a little snapshot of it. All right, questions on this? It's going to matter. All right, so let's start with the carbon cycle. It's just a little bit something you guys have all thought about more. And it's just a little bit simple. And I'm not going to make you learn every arrow on this in this course. There's other courses that make you learn every arrow, but not this one. All right? But what we're talking about here is from the perspective of a microorganism, how does carbon cycle through that environment and through an ecosystem? Okay? From the perspective of a microbe. I'm not talking about bunnies and lizards anymore and big beautiful trees. I'm talking about microscopic organisms. All right. And keep in mind, remember that microbes can live in an anoxic world, they can live in the absence of oxygen, and they can live in an oxic world, presence of oxygen. Incredibly enough, is so elegantly split in half as this is, they can also live on a gradient in between as well, right? Remember from your lab? So, let's start with the oxic side, just because that's what we all know and we're most comfortable with. So carbon enters the carbon cycle, is CO2 getting fixed into organic matter. And what is that process called? The answer is as easy as you think it is. No, the opposite of oxidation. Carbon carbon fixation. What is carbon fixation? What's the word you learned when you were in ninth grade for carbon fixation?
Photosynthesis?
Photosynthesis, thank you. Why was oxidation wrong?
Because oxidation, because that adds one more oxygen. CO2, CO2.
CH2O. So what have you done to that carbon atom?
Is that the carbon atom?
Yeah. From going from CO2 to CH2O. That's just a general term for carbohydrate. What have you done to that carbon atom? This is important. What'd you say, Green? Yeah. The, yep. Reduced it, thank you. You were really wrong, you just weren't hitting on what mattered most. You reduced it, so this is the process of carbon fixation is an energy expensive process, that's why plants need sunlight to get their electrons from water, right? Okay, so that's how carbon enters the carbon cycle, alright? And then things degrade that. Microorganisms, heterotrophic microorganisms will eat those sugars up and respire back CO2. So this first loop, all pretty simple and pretty understandable. You've thought about it in thousands of times in these various classes. Alright. You can also have it on the anoxic side. Let's jump over there. So carbon fixation can absolutely occur over there, and it does quite frequently, where an organism will take carbon dioxide and actively reduce it down to a sugar. There's plenty of cyanobacteria that can live in anaerobic environments and do this all the time. Alright, and so in so doing, CO2 turns into organic matter, just like over here it's in an anoxic environment. But it's the degradation of that organic matter in an anaerobic environment that gets a little different, all this kind of swirled, confusing arrows here. So in this process, in an anaerobic environment, you can take that organic matter, and believe it or not, in some sense, oxidize it to CO2 in a fermentative pathway. A lot of organisms will also just convert it to methane. And that's pretty much the scope of what you need to know for this class, because it dovetails in. If you're remembering the whole class well, it does tail in with the way I taught you other things. So you have, on the oxic side, carbon gets fixed into organic matter, gets respired back as CO2. On the anoxic side, you can have CO2 in some ways, used as a terminal electron acceptor, converted, CO2 converted to organic matter. And then you can degrade that organic matter through either anaerobic respiration, okay, something like dropping your electrons on nitrate, like we talked about before, or a fermentative pathway. So when sugars get degraded in a fermentative pathway and CO2 gets released, what are you making? It's one of your favorite things to do on the weekend, if you're your average, normal college kid. Drink beer. Absolutely. So this process of taking those sugars and converting it to CO2, no one wants a flat beer, you want a fizzy beer. Alright, that's fermentation, okay? And in a separate pathway, right here, going from organic matter to methane in an extremely anaerobic environment. So what other example have I given you that talked about that? Wasn't in this class. Yeah, the microbes that live in your gut. We're gonna spend some more time on that. Thank you. You weren't supposed to know that yet. That was another class. Microbes that live in the gut will absolutely convert the sugars that you consumed into methane, unfortunately. But you see this very heavily in cows, ruminants, organisms like that. Rice agriculture produces a lot of methane for these exact same reasons, because you're anaerobically degrading organic matter and releasing it as methane. Alright, so it's a big overview of the carbon cycle. And really just focus on this circle right here, and this circle right here, and then the production of methane. Alright, so let's just roughly go through quickly. Photosynthesis, it's the start of the carbon cycle again. And if you remember, it's capturing CO2. Alright. Going through the photosystems, capturing CO2 from the atmosphere as in the dark reactions, so many notes. Bear with me, okay? The light reactions, breaking up fuel the fixation of carbon dioxide to sugars, and that's the Calvin cycle. All right, so just tying this back to other things you've learned. You learned the Calvin cycle in another part of this class. What part of that class would it have been? You learned it before in here.
Synthetic bacteria.
Photosynthetic bacteria, absolutely. What did someone over here say?
It's a metabolism.
Metabolism, absolutely, good. So is this an anabolic or a catabolic reaction? Somebody else answer. Thank you. Anabolic, absolutely. It's anabolic because we're taking carbon dioxide and we're building something. We're inserting energy we're using precursor molecules, inserting energy to build bigger molecules. All right? So that's photosynthesis. That's getting carbon into the carbon cycle. Methanogenesis. So as I said, let's talk about it in a little bit more detail now. I jumped a little bit on my slides. Methanogenesis is something that goes on readily in anaerobic environments, extremely anaerobic environments. So very anaerobic environments that you might have thought of are like the gut of this cow or even your own intestines and farts. Okay, but certainly the gut of a ruminant animal like a cow, sheep, things like that is extremely anaerobic. And there's a lot of organisms in there, bacteria, archaea, and protozoa, that are breaking down the sugars that that animal eats when it eats grass. You ate that later.
Oh, shoot.
Another anaerobic environment that maybe you haven't thought about that much is something like flooded agriculture like rice fields, but flooded environments like the New Jersey Meadowlands. So who all has noticed that funky, glorious smell when you ride over the New Jersey Meadowlands? Okay, and what that is, in many cases, partly it's nested garbage heaps, which frankly, biologically is not that different than what I'm talking about here. But it's anaerobic metabolism in the sediments of that flooded landscape. The oxygen in the air can't penetrate down to that water as well as it can. It doesn't move through water as well as it moves through air. So you end up with sediments that have a whole lot of organic matter because you have rooted plants, be it either rice that's grown agriculturally or the roots of all those marsh plants out there, that Phragmites and Spartina and okay? You have a whole lot of organic stuff down there, rich organic, reduced carbon down there, sugars, etc., proteins, cellulose. Microbes are breaking that down like crazy without a lot of oxygen available. Okay, that releases a lot of methane. Another funky smell you get out of there is anecdotally, you also smell hydrogen sulfide, maybe you smell rotten eggs. That's also because they're using things like sulfate as a terminal electrons that releasing hydrogen sulfide. So if someone were to go walking through the Meadowlands and stir up all that sediment, it would release a lot of methane, release a lot of hydrogen sulfide, and get a nasty smell. Likewise, when that cow farts, he's releasing all those gases out of that environment. Okay. It smells great. Okay. So this is really important environmentally because methane is a hugely important greenhouse gas. We hear so much on the news about carbon dioxide as a greenhouse gas and its effects on the global climate. Methane is an even more potent greenhouse gas. It traps even more like energy. And so the microbial processing is going on in everybody's guts, as well as in flooded environments like rice paddies and things like that. So releasing a lot of methane, and that's part of the global carbon cycle problem that we're dealing with. But it's this process that you see here is strictly a microbial process. Either protozoa